So today I was going to take you through some of the work that we've just finished off, as well as a very brief overview of some of the work that we're moving on to now. The work that I've been doing since starting here has been taking over a grant that Chris was on, which is a DEFRA-funded project that works alongside the APHA. Since starting, I was mostly finishing off Objective 2, which was looking at the seasonality and overwintering of Culicoides, and I was part of a team, including Chris and Karen, that was looking at the transmission of bovine ephemeral fever alongside the APHA. I'm going to start with the overwintering of Culicoides. Laura's done a fantastic job of already explaining why we care about the overwintering and seasonality of these insects. So I'm going to move on to what we did. So Chris recruited 6 farms in Oxfordshire, 2 small, 2 medium, 2 large cattle farms, and we undertook OVI trapping on these farms from November 2016 to February 2017, and then from when I started in 2017 through to May 2018. These were weekly collections, and in total we made 220 collections throughout the project, which totaled over 6,500 midges that we speciated. We also have tiny tag temperature and humidity data. We've paired this with Met Office data from local sites around the farms, and myself and Marion last year undertook in-depth habitat surveys on all 6 farms to complement some farm characteristics that we were doing. We have since paired this with some of our national surveillance data overwintering that Marion has undertaken, and we've handed it all over to Simon Gubbins to hopefully find some results for us. To look at seasonality, we took 2 of these farms and we put emergence traps on the dung heaps. These are small tent-like structures that collect everything that emerges out of the dung. We ran these continuously from June 2016 to December, and then again from March through to May 2018. And we collect these every fortnightly, and we move them around to prevent suffocation of the environment. In total, we made 308 collections. This would have been more, but we had some interference from some cattle. Badger took particular offence to one of my traps, and one we lost to the dung, and it's dead to us now. This we caught over 96,000 Culicoides, and what was particularly interesting here was it was almost a complete monoculture. Over 90% of all of the Culicoides that we caught were Culicoides obsoletus, which is the vector of Schmallenberg virus and blue tongue virus here in the UK. And what was particularly interesting from collecting this monoculture was we saw quite a consistent emergence throughout the summer, and this seemed to correlate from preliminary analysis with increasing temperatures. But we were also seeing some sporadic emergence throughout the winter as well, and this is something that some of our later grants are going to kind of move on to. Again, we've paired this with Met Office data and handed it to Simon so he can do his modelling thing for us, because it's huge. The final objective was a bovine ephemeral fever transmission study. Bovine ephemeral fever is a rhabdovirus. It's actively circulating in the Middle East, Asia and Australia. It's pretty much what it says on the tin. It's also known as 3-day fever, and those are the typical clinical signs you would see in an outbreak in these areas. What's particularly interesting about this disease is that we know that it's probably spread by biting insects, but we don't actually know which biting insects. So this is why this disease was chosen for our coordinating capacity response grant. The APHA sourced an isolate from Israel, which was a cell-adapted isolate, and they created an assay, a real-time PCR assay, and looked at the mosquito side of things, whereas we took the midges. So to do this, we fed 250 of our 4-day-old Culicoides sonorensis from our colony here at Pirbright with a 1-to-1 blood virus mix. We left them for 8 days at 25 degrees, and then Chris separated head from body, and we homogenized these separately to determine not just replication but also dissemination to the salivary glands. And from this we found that we were getting about a 0.98% vector competence rate, which is similar to those observed in the blue tongue virus, serotype 8. The next step was going to be trying to reset this passage to 0 by sticking it in the midges and then feeding the midges onto the animals, because this virus does not seem to infect animals when we're using these tissue cultured strains. So we were hoping this kind of might be a little loophole. When we are going to feed infected midges on cattle, we obviously want a higher competence rate, because otherwise we're going to be feeding an awful lot of midges on these poor cattle. So Chris interthoracically inoculated Culicoides sonorensis. He interthoracically inoculated over 1,000 midges as part of this, and that was as hard to watch as it sounds, and I'm sure much harder to do. So Chris really pulled out the bag for this. We fed these after an incubation period on the cattle in the isolation units, and we undertook daily testing for bovine ephemeral fever in the blood. We also collected serum samples at intervals for antibody testing, and we got back pretty much absolutely nothing. Karen then challenged the animals again with both intradermally and subcutaneously, and we still got nothing. But what's interesting, and what we should take away from this, is the fact that we've got some first evidence that we've got vector competence in midges. No one else has managed to get this far, and we're very happy with that. We also played well with the APHA, and I think this is going to be a very nice paper at the end of it, even if it's mostly we did this and it didn't work. Moving on to what I'm doing now. This again had 3 main objectives, which I'm going to very briefly go through. Again, Laura touched on this earlier. Age grading of Culicoides is a bit of a problem. So, say I'm trapping in the winter in my OVI trap and I catch a midge. How old is that midge? Is it one of the ones that I've just seen emerging from my emergence trap, or has it been around a lot longer? And in an outbreak scenario, could it potentially be infected? Currently, we use parity pigmentation as a proxy for parity. Increasingly, this is being questioned as a reliable way of actually telling how old these midges are, and obviously it doesn't give you an actual time frame of how long this midge has been alive or how many feeds it's done. I'm currently creating a bank of midges of known age, so we can test 2 of the techniques that are already kind of being developed, which is the mouldy toff and NERS. I also have a student starting next month who's going to be doing some primer design for some milk curve analysis, which has shown some positive steps with mosquito age grading from wild field-caught mosquitoes. We'll also be doing another animal experiment, which is a time series feeding to define infectious period for blue tongue virus in the bovine host. And this year, again, I'm looking to try and find some local farms where we're going to get a known number of a lot of midges, because that's what we're going to need for this animal experiment in the following year. And the final thing that I'm up to at the moment is also recruiting farms for integrated vector trapping, and this we're focusing in on the larger biting flies. We're using a range of traps. These are just the photos I have. So we've got a horsefly trap, which is big and heavy and fun. We've got our normal midge OVI trap in the middle there, and that's one of the emergence traps. I'll also be using NZ traps, which are kind of not dissimilar to Malaise traps that you might have seen before. I'm currently recruiting different types of farms, and again, we'll repeat the temperature data collection as well as a habitat survey to be able to compare the differences between these farms, both in peak season, end of season, and across years. So that's the ultimate goal. So if anyone knows of any sheep or cattle farms that have a horsefly problem, let me know. So that's what I've been up to, and that's what I'm going to be up to. And I just want to say thank you very much to everyone that's been involved in the project, particularly Chris, who I've been massively reliant on, Karen, and Simon Govins, who has been dumped with all our data. So thank you very much.